So these are the various type of movements that you notice in case of organisms. So today's topic, what we are going to discuss is about locomotion and movement. So the locomotion is moving from place to place, and movement is also a part of locomotion. So they are helped by the skeletal system as well as the muscles. So we'll be going into details of what happens exactly during the locomotion and movement part of it. So let us begin with this topic. So locomotion is the voluntary movement resulting in a change in locomotion. You can see the muscles that are involved during a locomotion, like running, jogging, or galloping. I told you about horses gallop. We don't gallop. So the walking, running, and jogging. So you can see the skeletal systems that are also involved during movement. So the muscles as well as the skeletal system. So that is what the slide is about, to show you the involvement of muscles. Lot of bones are involved in that, the skeletal system and the muscles, the skeletal muscles associated with them. So they bring in about the locomotion. So locomotion is a voluntary movement resulting in a change in location. All locomotion are movements, but all movements are not locomotion. It is similar to all enzymes are proteins, but all proteins are not enzymes. Similar to that, all locomotion are movements, but all movements are not locomotion. See, locomotion and movement are interlinked. Example, in paramecium, they have cilia. So cilia help in the movement of food through cytopharynx. They guide them. So the movement of food through the cytopharynx is guided by the cilia. So you can notice the cytopharynx. It's almost mouth-like structure in this unicellular organism. So the cilia guide the movement of food through this cytopharynx. And in cilia also helps in locomotion. So you might find this ciliary movement. Hydra, they use tentacles to capture prey and for locomotion. So they help in capturing the prey and for locomotion. Limbs in us, like in most of the mammals, they are four-legged animals. So they do not have an erect posture like us. So they walk on their four. Whereas we use only the lower limbs for the locomotion. Limbs help to change the body posture and for locomotion. So the limbs help to change the body posture and for locomotion. So Hydra uses tentacles to capture prey and also for locomotion. So these are the tentacles. Even in case of octopus, you can see those 8 tentacles that helps them, and 8 arms rather. We can't call them as tentacles. The arms that help them in uh, movement as well as in capturing the food. Okay. I mean, the types of movement, what you notice is amoeboid movement. So in case of amoeba, you can notice that by pseudopodia. So in amoeboid movement, it is by the formation of pseudopodia or a false feet. Pseudopodia is nothing but false feet. So they are formed by streaming of protoplasm. So they are formed by the streaming of protoplasm. So the cytoskeletal elements like microfilaments within this amoeba also help for this. So amoeboid movement, see these types of movement in uh, human beings within the human cells. These are all not just in the microorganism amoeba. So here you can see within our body the macrophages and leukocyte. So these are types of movement within human beings. So there is amoeboid movement by the cells such as macrophages and leukocytes. So there is an amoeboid movement by the formation of a false feet by streaming of protoplasm. So the cytoskeletal elements like microfilaments within our cell, within the cells, they help for this amoeboid movement. Example is the macrophages. You can notice the macrophages are present in case of our areolar tissue. Okay, so they help in pseudopodia movement, the formation of false feet. The amoeboid movement is formed by this pseudopodia, which is streaming of protoplasm. So macrophages and leukocytes are example for amoeboid movement within our body. See, we might just look into the locomotion part of it, but there are movements. That is the reason all locomotion is not movement. We call it as, okay. All movement is not locomotion. So all locomotion are movements, but all movements are not locomotion. So ciliary movement by cilia. So here there are cilia in the trachea to remove foreign substances like microorganisms or any dust particles. So the cilia in trachea, they help in trapping the foreign substances. And oviducts, in oviducts also there are cilia for passage of ova. So they are helping, the ciliary movement is helping in removing the foreign particles in case of trachea or the windpipe, and in oviducts for passage of ova or egg. So the muscular movements by muscles. So this movement are by muscles. Example is movement of limbs. So the movement varies, the movement of limbs, tongue. See, when I'm talking, there is a movement of my tongue, jaws. The upper jaw is fixed, lower jaw is movable. So the jaws are moving. So these are all muscular movement. Muscular movement are brought about by muscles. Example, movement of limbs. I lift my hand. It is a movement of my limb. So it is forelimb movement. Okay. So tongue, whenever we are talking or whenever we are swallowing food. So there is a movement of tongue. Then jaws, the upper jaw is fixed to the skull, lower jaw is movable. So the lower jaw is movable when I'm speaking to you or delivering a class, isn't it? So this is the muscular movement. So you should understand about the third type of it, that is the muscular movement, which is brought about by muscles. Example is movement of limbs, tongue, jaw, etc. So we'll discuss each one of them in detail. So, amoeboid movement, I have already told you, it is the formation of pseudopodia by streaming of protoplasm. See here, the protoplasm is streaming and the pseudopodia is extending, streaming of protoplasm. So within them, they also have this cytoplasm has this elements like microfilaments, which also help for this amoeboid movement. So the white blood cells, you can see the leukocytes. So they are going to trap the foreign particles by this formation of pseudopodia. They also form the food vacuole and digest them. So within the blood, you can see the leukocytes. So they are going to uh, help in removal of this antigens or foreign particles like microorganisms by the formation of pseudopodia, which trap them and engulf them as a food vacuole. So the pseudopodia is going to form, and these two pseudopodia, they are going to fuse along with the microorganism, and they are pushed inside as a food vacuole and they are digested. So amoeboid movement is by pseudopodia, formation of pseudopodia, which is formed by the streaming of protoplasm. So the cytoskeletal skeletal elements like microfilaments also help for this. Example is macrophages and leukocytes. So the ciliary movement, you can see it in detail. The cilia in trachea. So there you find the microscopic cilia on the columnar epithelium, the inner lining of trachea. So these cilia help in removing the foreign substances. And cilia which are found in the oviducts, you can notice here the cilia is enlarged. They help in the movement of, and they are found in the oviducts, the cilia, which help in the movement of ova to the fallopian tube, where they are fused by these sperms. So inside fallopian tube, you can notice the guided movement of the ovum uh, for the passage of ova into the through the oviducts. Okay, so that is also brought about by cilia present in these oviducts. So the passage of ovum, so that is taking place within this oviduct. Still, they reach the fallopian tube. So that is by the ciliary movement. So it's a guided movement there. So this is inner lining of trachea and this is inside the fallopian tube. So how the cilia in the oviducts are assisting in the passage of ovum to the fallopian tube. So the types of movement in human beings. So I told you lifting, uh, running, jogging. See here, a person is doing a lot of exercises. Each of them are muscular movements, isn't it? So he's building up his muscles by the movement of muscles. So the muscles are built by the movement along with the weights that help them in building up of the muscles. Example, movement of limbs, tongue, jaw, etc. is also muscular movement. So within the types of movement in human, you also see the sperms, they will be having the long tail. Okay, so they have this flagellar movement, helps in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and in locomotion or protozoans like and in locomotion or protozoans like euglena, they also have this flagellar movement. So flagellar movement helps in the swimming of spermatozoa, maintenance of water current in the canal system of sponges. Okay, they have this choanocytes lining them. You know that water current water current in the canal system, water canal system of sponges, and in locomotion of protozoans like euglena, they have this flagellar movement. So the next part that we'll discuss is about the human muscular system. So the muscular system includes muscles which are all mesodermal in origin. So still now we have studied three types of muscles, isn't it? So the muscles which are smooth muscles or non-striated muscles. Then you have the striated muscles, or they are also called as striated muscles. Then third variety is the cardiac muscles. Okay, so we had discussed about these three types of muscles. All of them are mesodermal in origin. So during the formation of embryo, the mesoderm layer, it is going to is responsible for the development of muscles. So muscles constitute 40 to 50% of our body weight. So 40 to 50% of our body weight is by muscles. So muscles have this property of excitability, contractility, extensibility, and elasticity. So they have this, uh, muscles have this properties. That is, they are excitable, contractile in nature, they can be extensible and elastic in nature. So excitability, contractility, extensibility, and elasticity are the features of muscles. Based on location, muscles are of three types. The skeletal, striated, or voluntary muscles, they are under our control. So skeletal muscles, striated or voluntary muscles. Smooth, they are non-striated or visceral muscles. Okay. Cardiac muscles, they are found in the heart. So based on location, muscles are of three types. The types of muscles are skeletal muscles, visceral muscles, cardiac muscles. Skeletal muscles are striated or voluntary. The visceral muscles are smooth or non-striated, and cardiac muscles, which are again involuntary. So visceral muscles are also involuntary. So they are not under our control. So skeletal muscles are voluntary. So let us discuss about the first part of the skeletal muscles or striated muscles. They are attached to skeleton. Okay, so they are voluntary. So they are under our control. So that is why when you lift the weights, you can move the muscles, the biceps, triceps. They are all different regions of the muscles that can be built up. Okay, and they are voluntary in function. So they are attached. Skeletal muscles are striated muscles are attached to the skeleton. Striations are present in these muscles. They are voluntary in function. So they have a rich blood supply. So these muscles have rich blood supply. So these muscles undergo fatigue. So after a few moments or lifting of weights, so you get exhausted. So these muscles get fatigue or tired Okay, so muscle fiber is multinucleate, that is syncytium. So muscle fiber is multinucleate. So syncytium, that is there is no cross walls found. So more mitochondria is found in these muscles because they bring about the more respiration, they have more energy are liberated. So this is what you have to remember about skeletal or striated muscles or voluntary muscles. They undergo fatigue. Okay, if you look into the structure of it, you can see this outermost membrane, that is the sarcolemma, the anisotropic substances. The lighter region is the isotropic substance. The intermediate line you can see. Then there are myofibrils and nucleus. Syncytial because they are having multinucleate. So they don't have any cross walls. So they are multinucleate. So the muscle fiber, we can call it as syncytium since they do not have any cross walls, because of which they are multinucleate. The human muscular system, they have this visceral, or we also call it as smooth muscles. So in visceral organs, you find these smooth muscles. So smooth muscles are spindle shaped. They have a prominent nucleus. Okay, striations are absent. So these smooth muscles are involuntary. They are not under our control. You cannot control the movement of this visceral muscles or smooth muscles. So they have poor blood supply. So involuntary, they are having poor blood supply, the visceral muscles. They do not undergo fatigue. That is, they never become tired. Okay, they are uninucleate and less mitochondria, whereas the skeletal muscles or the striated muscles, they were multinucleate, whereas the smooth muscles or visceral muscles, they have lesser blood supply, they are uninucleate, and they have less mitochondria. So these are the features of this visceral or smooth muscles. The cardiac muscles, next we'll go into, they are found only in the heart wall. In the walls of the heart, you find this cardiac muscles. Striations are present. In cardiac muscles, you can notice these striations. They are involuntary. Cardiac muscles are involuntary. They are not under our control. They have rich blood supply. So cardiac muscles have rich blood supply. They do not undergo fatigue. They are non-fatigue. That is, they do not become tired. They are uninucleate, so they have more mitochondria. Okay, so that is what you notice in case of cardiac muscles. They are found why they should have more mitochondria, because they are responsible for the origin and conduction of impulses. So they are muscular movement of heart. The heart has to pump the blood, so that is brought about by the conduction and origin of impulses. So we have already studied about the sino-auricular node pacemaker, so which is the heart of hearts. So human being heart is myogenic. They are controlled by the muscles. The origin and conduction of impulses are controlled by the muscles. So they are muscular in nature. So the cardiac muscles should have more mitochondria because they require more energy to pump the blood to all parts of the body. So they are found only in the heart wall, the cardiac muscles. Striations are present, so they are involuntary. They are not under our control. You cannot control your heartbeat. So they have rich blood supply. So blood supply is rich. They do not undergo fatigue. That is, they are never going to be tired. So they are uninucleate. So you can see this branching of this cardiac muscles also you can notice, and they have this intercalated discs are present. So branching intercalated discs, so uninucleate, and they have more mitochondria. So these are the features of cardiac muscles. So the comparisons of muscles, all the three muscles, you can notice that the skeletal muscles are attached to the skeleton. Visceral muscles are found in the visceral organs. Cardiac muscles are found only in the heart wall. So skeletal muscle striations are present. Visceral muscles, they are absent. Striations present in cardiac muscle. So voluntary, visceral muscles are involuntary. cardiac muscles are involuntary. So skeletal muscles have rich blood supply, so poor blood supply in visceral muscles and rich blood supply in cardiac muscles. Skeletal muscles undergo fatigue, that is, they become tired. So there is no fatigue in case of smooth muscles, and even in case of cardiac muscles, they do not undergo fatigue or tiredness. So skeletal muscles are multinucleate since they do not have cross walls. They are, you can notice the syncytium with multinucleate condition. Visceral muscles are uninucleate, cardiac muscles are uninucleate. They also have intercalated discs. Cardiac muscles are also branched. Okay. More mitochondria is found in skeletal muscles, less mitochondria in visceral muscles. Cardiac muscles have more mitochondria. Next, we are going to discuss about is the structure of striated muscles. So you can see that there is a bundle of fibers. Within that fiber, there is another fiber, and they, when you dissect it, they are again having the fibers. Okay, so this is how the striated muscles they are. So let us study about the structure of striated muscles. See, skeletal muscles is made up of many muscle bundles. Okay, so skeletal muscles is made up of many muscle bundles. So we call it as fascicles. So this muscle bundles we call it as fascicles. So fascicles are held together by, see the fascicles, many fascicles they are having. So this fascicles, so the skeletal muscle is made up of many muscle bundles. So these muscle bundles we call it as fascicle. Fascicles are held together by the collagenous connective tissue layer, that is fascia. So the fascia holds them together. The collagenous connective tissue layer, that is fascia. Each fascicle they contain many muscle fibers. In turn, so each fascicle they'll be containing many muscle fibers. Okay, so many muscle bundles we call it as fascicles. They are all held together by collagenous connective tissue, that is fascia. So you can see this fascicle or muscle bundles and fascia. Then muscle fibers, which is also called as muscle cell. You can find each fascicle, they contain muscle fibers or muscle cell. So muscle fascicles. So you can notice here. So muscle bundles or muscle fascicles. So each bundle will be having the muscle fibers. So the muscle fibers they are lined by sarcolemma. So the muscle fibers are lined by plasma membrane, so which we call it as sarcolemma. So muscle bundle has this muscle fibers. Okay, you should understand muscle bundle will have many muscle fibers. So they are having this sarcolemma. Okay, so which encloses the sarcoplasm. So sarcolemma is the outermost membrane, and they'll be enclosing this sarcoplasm, that is the cytoplasm of the muscle. Each muscle fiber or the muscle cell or sarcomere, it consists of parallelly arranged myofilaments or myofibrils. So these are the parallelly arranged myofilaments or myofibrils. So the myofilaments or myofibrils, so each muscle fiber, they consist of the parallelly arranged myofilaments. So the outermost membrane is sarcolemma, the cytoplasm is the sarcoplasm. So each of this muscle fiber contains parallelly arranged myofibrils, which we call it as myofilaments, which we call it as myofibrils. Each myofibril has in turn, so you can notice each myofibril has a dark or anisotropic or A zone. You have this dark anisotropic, or we also call it as A band, and light striations, that is isotropic or I band. Okay, so isotropic or I band. So they have an anisotropic or A band. Each myofibril has alternate. So within the sarcomere, that is the cell, you find this. So each myofibril has a alternate dark anisotropic or A band and light striations, which we call it as isotropic or I band. So why is there a dark and light striations? This is due to the presence of two fibrous contractile proteins, actin and myosin. So actin filament proteins, thin actin filament. This is the thin actin filament and a thick myosin filament. So you can notice this thin actin filament and a thick myosin filament. So the dark and light bands are due to the presence of um, actin filaments, which are thin actin filament, and dark bands, they'll have this thick myosin filaments. I bands contain actin. Anisotropic band, they contain both actin and myosin. So that you have to remember. So I band or isotropic band, they just contain actin, whereas anisotropic bond, they have both actin and myosin. Okay. And you can see in the anisotropic band, actin and myosin are arranged parallel to each other. Okay. So the A band or anisotropic band, they bear a lighter middle region called H band. So they bear a lighter middle region called H band. So this H zone is a lighter middle region called as H band. So it is formed of only myosin. See the light bands or isotropic bands, they are made up of only actin, and in case of the anisotropic band, they are made up of both actin and myosin. Myosin are thick in nature, thick filaments. So the H band, it is formed only of myosin. So remember that. So A band bears a lighter middle region. Anisotropic bond, it has a lighter middle region, and you cannot see any actin filaments here. So they are made up of only myosin. A thin dark line, M line, runs through the center of head zone. A thin dark line or M line, excuse me, they run through the center of head zone. A thin dark line or M line runs through the center of head zone. So this you can understand. So I band, isotropic band, they'll be having only actin filaments. Anisotropic bands, they'll be having both actin and myosin filaments. So they have a thick myosin deposition. And in between the I band, there is a lighter middle region called as H band. So it is formed only of myosin. And this myosin also has a thin dark line called as M line, which runs through the center of H zone or the H band. It runs through the center of H zone or H band. High band is bisected by a dense dark band called Z line. So I band, that is the isotropic band, is bisected by a dense dark band called as Z line. So can you look into this Z line? So dense dark band called Z line. So the I band, that is the isotropic band, is dissected by Z line. So anisotropic band is dissected by this M line. So the Z line, M line, Z line. So region between two Z lines is called sarcomere. The region between two Z lines, we call it as sarcomere. So the region between two Z lines is called sarcomere. Sarcomere is the structural and functional unit of a muscle. So sarcomere is the structural and functional unit of a muscle. So the skeletal muscles, they are made up of muscle uh, bundles. So these muscle bundles are in turn made up of many muscle fibers. So the muscle fibers, so they are called as sarcomeres. They have the outermost plasma membrane called as sarcolemma, and they have sarcoplasm. So the muscle cells, they would be having parallelly running myofibrils. or myofilaments, we call it as. When we see this myofibrils or filaments, we find the light band. It is made up of isotropic. Light band is also called as isotropic band. They are made up of only actin, and the dark band, they are also called as anisotropic band. They are made up of both actin and myosin. So within this anisotropic band, at the middle region, they are lighter, and you can notice that they are made up of only myosin. So this lighter zone, we call it as H band or H zone. So there is a line passing through the center, which we call it as M line. So also we can notice in this isotropic band, there is a dense line. So I band is also, isotropic band is also bisected by a dense dark band, which is called as Z line. So the region between two Z lines, we call it as sarcomere. What are sarcomeres? They are the structural and functional unit of a muscle. Sarcomeres are the structural and functional units of a muscle. So you can notice here, so the different things what I have discussed with this diagram. So here Z line is there, then it head zone, which has a middle line, which we call it as M line. So M line is made up of only myosin. So M line is there. So H band is made up of, or H zone is made up of only myosin. So I band is dissected by a dense dark band called as Z line. The region between two Z lines, we call it as sarcomere. Sarcomeres are the structural and functional units of a muscle. Now let us understand the structure of contractile proteins. So actin and myosin are the contractile proteins. What is actin? Actin, an actin filament is made up of two filamentous F actins which form double helix. So an actin filament is made up of two filamentous F actin, we call it as two filamentous actins. An actin filament is made up of two filamentous actins, which is also called as F actin. Can you notice here the green structured color? So they are helically bound, just like DNA, they are helically bound. So an actin filament is made up of two filamentous actins, or we also call it as F actins. F actin, which form a double helix, you can notice that they are forming a double helix. F actin is a polymer of monomeric globular actins. So the filamentous actin is a polymer. So you can see many of the structures. It's a polymer of monomeric globular actins. So these are all globular actins. F actin or the filamentous actin is made up of a polymer of monomer globulins or globular actins or G actins, you can call it as. F actin is made up of, it's a polymer of many monomeric G actins or globular actins. So actins contain two other proteins, namely troponin, tropomyosin. You can notice this is like a thread it is wrapped around. Tropomyosin. Actin contains two other proteins, namely tropomyosin and troponin. Troponin are small structurals that you can notice, tropomyosin and troponin. So two filaments of tropomyosin run along the grooves of the F-actin. So there are two filaments of tropomyosin which runs along the grooves of the F-actin double helix. Troponin has three subunits. It is located at regular intervals of tropomyosin. Troponin has three subunits. They are located at regular intervals of the tropomyosin. In the resting state, a subunion of a subunit of troponin, a subunit of troponin may mask the binding sites for myosin on the actin filaments. So that is about the actin filament. So actin is made up of uh, two helically coiled uh, F actins or filamentous actins. Filamentous actins have this, they are a polymer of monomeric globular actins or G actins, we call it as. Okay. So the actin contains two other proteins, that is tropomyosin, which is also helically two filaments of tropomyosin are there, and they run along the grooves of the F-actin double helix. So troponin has three subunits. It is located at regular intervals uh, on the tropomyosin. In the resting state, this a subunit of the troponin, they mask the binding site for myosin. So the subunit of the troponin, they are going to mask the uh, binding site of the for myosin on the actin filament. Okay. Next, we'll discuss about is the myosin. So still now we discuss the structure of the actin. Now the structure of another contractile protein, myosin. So each myosin filament is a polymer of many monomeric proteins called as meromyosins. So each myosin filament is a polymer of many monomeric proteins, which we call it as meromyosins. Remember it as meramyosin. Okay, so meromyosins. A meromyosin has two parts. So we have to remember this through this structures. A meromyosin or meramyosin has two parts. The heavy meromyosin or HMM and the light meromyosin or LMM. So heavy meromyosin or HMM, so they are cross arm. So they have this globular head plus short arm. So the heavy meromyosin has a globular head and a very short arm. It projects outwards. It is just like the hood of a snake. So it projects outwards. So you have the meramyosins. One is the heavy meramyosins and light meramyosins. So remember it for fun, or it becomes very boring, isn't it? So heavy meramyosins instead of meramyosins, and light meromyosins. Heavy meromyosins, they have two parts, the head region and a short arm, and it projects outwards just like a snake would. Light meromyosins or LMM is the tail part of it. So each myosin filament is a polymer of many monomeric proteins called meromyosins. A meromyosin has two parts, the heavy meromyosins, which has a head region and a short arm. So globular head and a short arm. It projects outwards. The light meromyosins or LMM tail. So they form the tail region. The globular head of the heavy meromyosin is an active ATPase enzyme. So the globular head is an active ATPase enzyme. It has an is an active ATPase enzyme. So it has an binding site for ATP. It has a binding site for ATP and active site for actin. So it has binding site for ATP and an active site for actin. So the actin binding site, and they have this ATP binding site. The globular head is an active ATPase. It has an active ATPase enzyme. So here what you can notice here above is the actin filament with the troponin, three subunits of troponins which mask this actin also. Then below you are finding the meromyosins. So myosins are made up of polymer of meromyosins. Meromyosins, the heavy meromyosin you can notice with the head region and a short tail. And the tail region of the meromyosin is a light meromyosin which forms the tail region. So they are projected above. So you know about this. So these meramyosins have actin binding site as well as they also have this ATP binding site. So they are made up of this ATP synthase enzyme. Okay, the mechanism of muscle contraction we'll understand. Once you have understood the structure of actin and myosin, okay, we will understand the next aspect, that is the mechanism of muscle contraction. According to sliding filament theory, the contraction of a muscle fibers occurs by the sliding of thin filaments. You know, the thin filaments actin over the thick filaments, they slide over. I think you can notice that sliding filament theory, whenever there is a contraction of a muscle fiber occurs, by the sliding of the thin filaments, actin, over the thick filaments, myosin. Myosin remains as it is. So the sliding of the thin filaments, actin over the thick filament, myosin takes place. So whenever there is the sliding, so this theory we call it a sliding filament theory. So whenever there's a contraction of muscle fiber, the contraction of muscle fiber occurs by the sliding of the thin actin filament over the thick filaments, that is the myosin. So an impulse from central nervous system reaches the neuromuscular junction. So motor end plate via motor neuron. So neuromuscular junction is synapse between neuromuscular junction. It's a synaptic region between a motor neuron and sarcolemma of muscle fibers. So the synaptic region between a motor neuron and the so the synaptic region between a motor, the neuromuscular junction, here they have boxed it up. You can see that it is the synapse between a motor neuron and sarcolemma of muscle fiber. A motor neuron plus muscle fiber equals a motor unit, we call it as. A motor neuron plus muscle fiber, so we call it as a motor unit. So that is an impulse from CNS, the central nervous system, reaches the neuromuscular junction via motor neuron. So it is going to reach, an impulse is received from CNS, central nervous system. It reaches the neuromuscular junction via motor neuron. Okay, that is the first step. The synaptic vesicles release a neurotransmitter acetylcholine. So the synaptic vesicles, they are going to release a neurotransmitter, that is acetylcholine. So synaptic vesicles, this is the synaptic knobs of this motor neuron. They are going to release a neurotransmitter acetylcholine. So which generates an action potential in the sarcolemma that spreads. So this neurotransmitter acetylcholine generates an action potential in the sarcolemma. So there is the junction between the muscle and the neurotransmitter, that is the neuromuscular junction. So synaptic vesicles, they are going to release a, yes, this is the synaptical vesicles which release the neurotransmitter acetylcholine. So acetylcholine generates, so the action potential, so this is the enlarged part of it, action potential in the sarcolemma, that is the plasma membrane of the muscle that spreads through the muscle fiber. So once there is a this action potential that is created by the sarcolemma, that is created on the sarcolemma, so it causes the release of calcium ions from sarcoplasmic cisternae. So they would have labeled it here. So the release of the calcium ions from sarcoplasmic cisternae into sarcoplasm. So that is going to take place. So there is a, once the action potential spreads through the muscle fibers, there is a release of calcium ions from sarcoplasmic, that is cytoplasm of muscle fibers, sarcoplasmic cisternae into sarcoplasm, that is cytoplasm of muscle. So calcium binds, see the calcium ion binds with a subunit of troponin. So there is a tropomyosin and troponin. So the calcium binds with subunit of troponin and unmasks the active site for myosin. So this is going to be the, it is going to unmask the active site for myosin. Okay, using the energy from ATP hydrolysis, that is the breakdown of the so there is the actin filament and there is the calcium ion which is unmasking the active site for myosin on this subunit of troponin. So using the energy from the hydrolysis of ATP, breakdown of ATP, the myosin head. So you can notice this myosin, mera myosin head, that is heavy mera myosin, HMM. So the myosin head, using the energy from ATP hydrolysis, the head binds to the active site on the actin. So it binds to the active site on the actin. So which is the subunit of troponin formed by the uh, unmasking the active site for myosin. So calcium is going to bind with subunit of troponin on the actin filaments and unmask the active sites for myosin. So the myosin binds to this active site on the actin to form a cross bridge. Can you notice here it binds to the myosin, actin binds to the myosin to form a cross bridge. This pulls the actin filaments on both sides towards the center of A band. So this pulls the actin filaments on both sides towards the center of A band. So actin filaments partially overlap. So they are going to partially overlap so that the H zone disappears. So the actin filament partially overlap. You know that H zone had only myosin filaments, but because of this cross bridge forming and the pull of this actin filaments on both sides towards the center of A band, so the actin filaments partially overlap so that H zone disappears. So this is what is happening. So the here you know the sliding filament theory. So the H zone that is there before contraction in the relaxed state. So whenever the myosin is moving over because of this, okay. So the this pulls the actin filaments on both sides towards the center of the A band, so center of anisotropic band, and you can see that H zone disappears. The Z line attached to actins is also pulled inwards. So Z line, you know that this Z line is also pulled inwards, which is attached to the actin. It is also pulled inwards. It causes the shortening or contraction of sarcomere. So whenever there is contraction, it causes the shortening of sarcomere. Okay. So I band gets shortened and A bands retain the length. So I bands get shortened and A bands retain the length. So that is happening during the movement of muscles, contraction of muscles. Now the myosin releases the ADP and inorganic phosphate and goes back to its relaxed state. So the myosin releases the ADP and inorganic phosphate and goes back to its release state. A new ATP binds and the cross bridge is broken. So the ATP is again hydrolyzed by the myosin head and the above process are repeated causes further sliding. So you should first understand the structure of actin. You should understand the structure of myosin. You should know what is isotropic band made up of. Isotropic band is made up of only actin filaments and they have a line in them that is called as Z line in between them. So the isotropic bands are divided by the uh, Z line. So the distance between two Z line is called as uh, the region between two Z lines is called as sarcomere. Then there is the anisotropic band or dark band. So they are made up of both actin and myosin filaments. So they have an intermediate at the center of this anisotropic band. They have a, uh lighter zone, which we call it as H zone. So or H band. So through the center of which you can find the M line is there. So now the H zone doesn't have actin, but it has only myosin. Okay, so actins are thin filaments, myosins are thick filaments. So that you have to understand. So whenever there is contraction, and when you understood the structure of actin, you know that they are having this two F filamentous actin. So F actins, they are helically coiled, and along with this helically coiled, two helically coiled tropomyosin is there, and they have at regular intervals three subunits of troponin uh, present. Okay, so tropomyosin and troponin which are present. Troponin, one of the subunit of troponin is going to unmask the active binding site of actin. So that is what they are going to do. So finally, after this contraction, the disappearance of H zone. So the myosin, once it releases, see they have pulled it, uh, they are pulled back. So the sliding is happening and the breaking of cross bridge takes place. So there is a cross bridge formed. So that breaking of cross bridge takes place once ATP is synthesized. Okay, myosin releases the ADP and PI inorganic phosphate and goes back to its related state. A new ATP binds and the cross bridge is broken. The ATP is again hydrolyzed by the myosin head and the above process are repeated, causing further sliding. So this is the sliding mechanism that we are talking about. So the actin myofilament will be have the active site on which the head region, the heavy meromyosin is going to bind. So once the myosin head binds with this actin filament, that is they are going to form a cross bridge. So the cross bridge, they are going to slide or rotate. Okay, and after the sliding or rotation that is happening because of the contraction of the muscles. Okay, so the eye bands get shortened and you can notice that the eye bands get shortened and the Z band, A band retains the length. Okay, so this is the relaxed state where you can see the H zone, high band is there, A band is there. So in the contracting zone, you find that the uh, the anisotropic bands, there is no change, but the I band, they are going to get shortened and the A bands retain the length. Okay, so the maximally contracted. After that, they get back to their original position. When the myosin releases the ADP and inorganic phosphate and goes back to its relaxed state, a new ATP binds to the binds and the cross bridge is broken. So the ATP is again hydrolyzed by the myosin head and the above process are repeated. So the ATP first should be hydrolyzed by the myosin head, that is, they should be broken down. Only then they can get the myosin head can get attached to the active site of the actin and they form a cross bridge. Okay, so this is the sliding theory to explain the muscular contraction in case of the mechanism of muscle contraction. So that is explained by this sliding filament theory. So contraction of muscle fibers occurs by the sliding of thin filaments actin over the thick filaments. Slow sliding of the thin filaments actins over the thick filaments myosin. So that sliding filament theory is what explains the mechanism of muscle contraction. Okay. So you have to write this 10 points step by step, and that will help you to score the marks. So this diagram is what is given this, and this diagram is given in your textbook for you to understand. In the examinations, they are very rarely asked, but you should understand these mechanisms. Those students who are planning for your medical examinations, you are planning to take up medicine, so you should be well versed with this concept because even at higher levels in your medical classes, so these concepts would be helping you to understand better. Okay, so this 10 points you have to remember. So the when calcium ions are pumped back to the sarcoplasmic cisternae, calcium ions initiated everything at the beginning, isn't it? So when calcium ions are pumped back to the sarcoplasmic cisternae, the actin filaments are again masked. So the unmasking of this actin filaments are done by this calcium ions, which bind to the one of the subunit of troponin. So as a result, Z lines return to their original position and it results in relaxation. So when calcium ions are pumped back to the sarcoplasmic cisternae, the actin filaments are again masked. As a result, Z lines return to their original position and it results in relaxation. So the next is mechanism of muscle contraction. Some points that you have to remember. The reaction time of the fiber varies in different muscles. It might not be the same. Repeated activation of the muscles leads to the accumulation of lactic acid. So there would be anaerobic respiration in the muscles, and that leads to the accumulation of lactic acid. Repeated, you're lifting some weights. So you're doing it 10 times, 12 times, repeatedly you're doing it. It leads to the accumulation of lactic acid, and it causes, whenever you undergo fatigue, it causes muscle fatigue because of the accumulation of lactic acid. This is due to anaerobic breakdown of glycogen in muscles. I told you that 500 grams of glycogen is stored in our body. It is just like reserve, just as in your bikes, reserve fuel is there. Similarly, we have reserve food stored in our body. 100 grams is stored in liver as glycogen, and in our muscles of our body, 500 grams of glucose is stored as glycogen. So they are the reserve food material. So the whenever we are doing repeated activation of the muscles, you're overstraining yourself, you're running, jogging, lifting weights. So what is going to happen is repeated activation of the muscles, any of these muscles, it leads to accumulation of lactic acid, which causes fatigue. The muscles become tired. You can't do any more exercises. This is due to, why is this happening? Why is there a fatigue of our muscles? This is due to anaerobic breakdown of glycogen stored in our muscles as lactic acid. Okay, so that is why our muscles undergo fatigue. This is due to anaerobic breakdown of glycogen in our muscles as lactic acid. So red muscle fibers and white muscle fibers. So you should understand about the differences between it. Red muscles, they have more mitochondria. Mixed muscles, they have medium mitochondria. White muscles, they have less mitochondria. So these are the muscle fibers we are talking about. Red muscles, they are having more mitochondria. Mixed muscles, they have medium mitochondria. White muscles have less mitochondria. So red or aerobic muscle fibers, red color due to myoglobin. Okay, the muscles are red color due to myoglobin. White muscle fibers are white color due to lesser myoglobin. So red muscle fibers have more mitochondria. White muscle fibers have less mitochondria. Okay, aerobic metabolism takes place by the red muscle fibers. Anaerobic metabolism takes place in the white muscle fibers. So red muscle fibers are slow and sustained contraction. White muscle fibers are fast contraction for short period. So sprinting, running for 100 meter, so you'll be using more of your white muscle fibers. And if you are running a marathon, you are using the red muscle fibers. So slow and sustained contraction, red muscle fibers. White muscle fibers, they are for fast contraction for short period. The human skeletal system, we'll discuss about it in the next class. So today's class, we have discussed about the sliding filament theory for mechanism of muscle contraction, then differentiated between red muscle fibers and white muscle fibers. We also studied three types of muscles, that is the skeletal muscles or striated muscles or voluntary muscles, then visceral muscles or smooth muscles or non-voluntary muscles. Then we studied about cardiac muscles, which is again involuntary muscles. So these are the topics that we covered in today's class. Any doubts or clarification, dear students? So please watch this recorded video also again. So because the muscle contraction theory, you have to understand it step by step. Okay. So any doubts or clarification, dear students? So today